This next potential energy surface is a much more complicated one than the previous one. It comes out of some research from the Stahl group that is well known for their work with organometallic catalysis as well as other things. Um, it's going to illustrate some very important concepts and is a little bit simpler if you just focus in on a couple of pieces. It represents a reaction pathway from an acyclic amine species here, reacting with a chiral palladium catalyst, goes through a whole lot of transformations. Some of the steps I haven't even shown and I don't really want to focus on here. I want to focus on the fact that out of this reaction, they observed only the product that's labeled as S over here, the black one. Um, Only one isomer came out of this reaction, which is pretty impressive. They started with an uh, acyclic species, a chiral catalyst, and got a very selective reaction that produced only one of the cyclic amine species. Now, you actually only need to know sort of the stuff we learned in Gen Chem in order to be able to make sense of this with one new piece of information. And I want you to put this number in your head for the entire Chem 344 semester. Essentially, you can compare the relative abundance of two species just by looking at their energy difference. And an energy difference of about 1.4 kcals roughly translates into a factor of 10 difference in their abundance. And that plays a big effect right here. To the extent that these numbers are accurate, and the method used by this group to model the reaction is meaningful, there is a roughly 3 kcal per mole difference between the energy barrier needed to get over the reaction pathway for the blue unobserved isomer versus the black actually observed isomer. And this is sort of the key moment. As we learned in Gen Chem, the most important moment in any reaction is the highest energy barrier, the slow step, the rate determining step. And that moment happens right here in this reaction. And since the lower pathway is favored by 3 kcals over the upper pathway, that translates into, well, 3 divided by 1.4 is like 2. So about a factor of 10 squared, or 100. It is 100 times more likely to go over this bottom pathway than this top pathway. Well, if you favor one pathway by a factor of 100, that means greater than 99% of the product will be the favored path, follow that favored pathway, and lead to the favored product, and only less than 1% will lead to the other isomer. So this is a huge deal. And really what you need to focus on is that a small difference in the energy of the highest energy barrier can lead to a huge difference in the observed outcome of a reaction. And so this is some work that I thought was interesting and came out of the Stahl group and gives you a nice understanding of a potential energy surface. Also coming out of my research group, and this work was done about 8 years ago now, looked at a reaction of two really weird organic species. These are both molecules of astrochemical relevance. A lot of the work in the McMahon group sort of reflects that. I think these are really neat species because they're not easy to investigate because they're so reactive, they're so missing hydrogens and unsaturated that these need to be isolated from each other, isolated from any reactive molecules around it, and kept at temperatures on the order of 10 to 25 Kelvin, which is pretty damn cold. And as a result, you really can't use sort of the traditional methods that organic chemists use to investigate structures. We rely on matrix isolation techniques and using IR spectroscopy. A researcher in my group in the past investigated this reaction and did so by IR spectroscopy. And essentially, the data comes out like this. This is a subtraction spectrum. Computational chemistry, which is absolutely critical to this work, was used to confirm that the top red spectrum here is the product, and the bottom blue spectrum here is the calculated spectrum of the reactant. These calculated spectra are used to confirm the experimental spectrum right here. It turns out that any line aimed downward in subtraction spectra is something that was reacted away. So we should be able to see that all of these downward facing peaks line up with the ones predicted, showing that this reactant was reacted away as light was shined on it. And in the other case, we see these peaks growing in, supporting that this product was formed um, as this species reacted away. So essentially, he was able to convert this thioketene to a diacetylene thiol species by shining light on it, which is a pretty neat reaction. Both of these molecules have a fair chance of being found in the interstellar medium and are sort of important molecules in that regard. A final research example that I wanted to give you a flavor of is some really neat work I think that could take place in the next couple of decades as we start to get faster and faster computers and better modeling software relating to proteins. Um, right now, we don't have good enough computational resources to model the folding of a protein and even allow the pocket of a protein to sort of adjust as a molecule is placed in that pocket. But as our computers become stronger and stronger in terms of their computational capability, we might see this type of calculation I'm about to go through just really take off in the next couple of decades. Essentially, this is some work coming out of the Kiesling group where they were looking at how a natural molecule might bind itself in the reactive pocket of a protein. And so we know the structure of the protein from some crystal structures of proteins, and we have information about what sites on the molecule are as the active pocket. We can use a computed structure of a molecule we know binds to that pocket, put it in the pocket, and the computer is able to sort of figure out how it's held in place. In this case, there's some green dotted lines sort of showing how that's held, and there's some hydrogen bonding interactions that keep that molecule held into place. One of the interesting bits about this type of research is the member of the Kiesling group doing this work was able to take this unnatural cofactor with a fluorescent group attached to it and put it in the same pocket of the protein and see if it was able to bind. And they were observed that it made some of the same sorts of interactions like here, but different hydrogen bonds actually held it into place. And so that's kind of a cool way where they can look at different structures and see how they might or might not bind to a particular protein active site. So that's an area of research that I think is going to get increasing attention in the next couple of decades as our computers are better able to handle these pockets and start to allow the protein itself to change conformation and to fold around the cofactor as we know they actually do. Right now, the pockets are kind of static, and so this is something that's still a growth area of computational chemistry.